So today I'm going to be talking about the most distinctive and iconic of marine fish genera, the seahorse genus Hippocampus. More specifically, I'll focus on the hidden biodiversity in seahorses from Australia and the Indo-Pacific. Before I start, I'd like to thank the Australian Museum and AMRI for giving me the opportunity to help me advance my research on this group of fish. And I would also like to mention this project has been partially funded by the AMRI Visiting Collection Fellowship. So presently, only a single genus is currently recognized for all seahorse species. The absence of a clear separation based on external features, even among the more distinctive species that you see here on this slide, makes it currently difficult to justify the recognition of one or more genera. Most species are superficially similar, which leads to challenges in identification and especially conservative priorities, since many species are listed as threatened on national and global red lists. Out of the entire animal kingdom, male seahorses are the only males in the world that undergo pregnancy and give birth to their offspring. The eggs are carried in a specialized brood pouch, which functions in a similar manner to the mammalian uterus. They also live in some of the most vulnerable inshore marine habitats, such as seagrass, estuaries, and coral reefs in temperate and tropical regions. Whoops. So, seahorses are members of the family Syngnathidae, which also includes the pipefish, pipehorses, seadragons, and pygmy pipehorses. The closely related ghost pipefish at the bottom left is closely related to the family. These Syngnathidae groups are united by their tube-like snouts, bony plates in place of scales, reduced number of fins, and of course, male pregnancy. This family is comprised of 57 genera and over 300 described species in marine waters. The biodiversity of Syngnathids in Australia is really, really high. There are 93 species and 47 genera that occur here, and amazingly, 54 species are endemic. Is all okay over there? You guys can hear me?
Yeah, all good, Graham.
Something happened with the connection there. So continuing on with the biodiversity, there are 93 species and 47 genera that occur in Australia, and amazingly, 54 species are endemic. 36 species are endemic in southern Australia alone, which is unbelievable. And in contrast, neighboring New Zealand has a total of 8 species, 4 of which are endemic. Seahorse species diversity, 43 species of seahorses occur worldwide, with 18 species occurring in Australia. Of those 18 species, a whopping 10 are endemic to Australia, which amounts to around 22% of all seahorse species. 6 endemic species occur in southern Australia alone, the remaining 4 in northern Australia. And now let's compare these numbers to other regions in the world. Only 5 species occur in North and South America combined, whereas only 11 species occur in the North Atlantic, Mediterranean, and Africa. And lastly, 15 species occur throughout the Western and Central Pacific Ocean. So you can see that seahorse biodiversity in Australia is really, really amazing. So in 2017, Healy Hamilton and co-authors published a study on a multi-gene phylogeny to investigate the relationships of Syngnathids at the generic level. As part of the study, and over the course of many years, we collected many rare species that were not available for genetic analyses from museum fish collections. As a result, we built up one of the most comprehensive collections of Syngnathids for tissue subsampling, which are now housed at the Australian Museum and the Cal Academy. One of the burning questions we wanted to resolve was which species will be recovered as sister to the seahorses. Our hypothesis, which was based on 4 nuclear markers, recovered a diverse group of Syngnathids as sister to the seahorses. This group is comprised of a clade of Australia occurring and endemic pipefish that you see in the orange, as well as a clade of Indo-Pacific pygmy pipehorses highlighted in light blue. Pygmy pipehorses are very similar in appearance to the seahorses since they have an angled head and prehensile tail and are often misidentified as seahorses. Most importantly, the phylogeny also recovered sampled members of Hippocampus in a morphological clade in which the seahorses clustered into 2 distinct genetic lineages, the non-pygmies and pygmies that you see here in the red, with a notable evolutionary distance between these 2 groups. These two lineages within Hippocampus strongly indicated to me that the genetic data and the current classification of Hippocampus need to be reconciled based on a more comprehensive morphological taxonomy. Fortunately, many rare species of seahorses, especially pygmy seahorses, are housed at the Australian Museum, which contributes significantly to my study. So before I get into the morphological data, I'm going to focus on the seahorses that represent these two lineages. The non-pygmy seahorses are comprised of 38 species that range in size from 2.3 to 35.5 centimeters in length. They're all characterized by the presence of a coronet-like structure on their head, a tubular snout with a fused jaw that enables suction feeding, and two gill slits, each of which are located on the other side of the head, a male brood pouch, which is located on the abdomen or trunk, and finally, prehensile tail. As you see here, Hippocampus abdominalis from Australia and New Zealand is the largest species at 35 centimeters. Hippocampus australis on the right from Western Atlantic reaches a length of only 5 centimeters and is considered a dwarf seahorse species. But it's still a non-pygmy seahorse. Hippocampus debilius from the Red Sea in the middle is the smallest species at 2.3 centimeters and is found in soft coral. This tiny seahorse species overlaps with the small sizes of the pygmy seahorses, but it's also considered a dwarf seahorse due to the presence of 2 gills and a male brood pouch. In comparison, the pygmy seahorses are comprised of 8 species and occur throughout the Indo-Pacific, 3 of which occur in Australia. They range in size from only 13 millimeters to 25 millimeters in length. They're also characterized by the presence of a coronet-like structure, tubular snout with a fused jaw, and a prehensile tail. They differ mainly in having only 1 gill opening located directly behind the coronet, and 2, the absence of a male brood pouch. Instead, the eggs are brooded within the trunk of the male. Pygmy seahorses can be grouped by their preferred habitat. In the first row above there, Hippocampus Denise and Bargabanti live exclusively in Gorgonian coral. The second row, Hippocampus culmini, Tapapigu, Nalu, and Pantohi, are free living and are found on sand, bryozoans, and halimina. And the third row, Hippocampus satomiae and Wellinanus, they live in association with soft corals. So, how do we reconcile the genetic data with the existing classification of Hippocampus based on existing morphological characters? As shown before, the non-pygmies and pygmies share many obvious external features, such as the coronet, angled head, and prehensile tail. But it appears that the few external differences between them, such as the size and the number of gills, are not sufficient enough to reconcile this data. So, therefore, new morphological characters are needed that are informative and reliably diagnostic. Thanks to Adam Summers at the Friday Harbor Marine Labs in the state of Washington, I've been using a powerful tool to examine the skeletal features of seahorses over the past few years for new species descriptions, such as Hippocampus tapapigu and Hippocampus nalu. That tool is microcomputed tomography, or CT, a technique that bombards specimens with X-rays to ultimately produce 3D images, and which can easily discern several features. Investigation of seahorse specimens from the Australian Museum and elsewhere revealed a number of really interesting morphological differences in the skull or neurocranium between the pygmy and the non-pygmy seahorses, and even among the pygmy seahorses that can assist in the reconciliation of genetic data. I'm going to play a little scan here for you that shows the fine detail. Oops, it's not playing here. Of the skeleton of a seahorse. You can see how ossified the head, tail, and trunk are. You can see the eggs within this pregnant female of this paratype of Hippocampus culmini. So, let's see here. It turns out that the morphology of these neurocranial characters, so in the neurocranium of these pygmy seahorses, I found a lot of variation. And these neurocranial characters are just as informative as skeletal characters typically investigated in other fish families, such as the primary or pharyngeal jaws in gobies and wrasses. So this was the key to be able to distinguish between non-pygmy and pygmy seahorses. And now I'm going to take you down the rabbit hole of seahorse taxonomy. I'm going to provide a brief lesson on the bones that form the neurocranium and which characters I consider diagnostic. This is a CT scan of the non-pygmy seahorse Hippocampus camelopardalis from Tanzania. As you can see, the head, trunk, and tail are heavily ossified. This is typical for all non-pygmy seahorses. There are 4 bones in the neurocranium that I consider to be diagnostic. A, the supraoccipital or SOC, the anterior nucleoplate for B, which is in fact highly modified to form the coronet that you see in seahorses. C, the posterior nucleoplate, and D, the clithrum. On each side of the head, the clithrum is discontinuous, where each gill is located in the soft flesh over this bone. So here in non-pygmy seahorses, the presence, arrangement, and morphology of these bones are typical for all non-pygmy seahorses. You can also see that these bones are in contact with each other. As for the pygmy seahorses, I've observed strong morphological differences in the morphology of their neurocranial bones, which just reinforce how different they are between the 2 lineages. So I'm going to start with the free-living pygmy seahorses. These are active species which were all described in the 2000s and have been observed on sand, seagrass, bryozoans, or the green macroalgae Halimeda. All are similar in appearance and appear to have wing-like structures on the first and second trunk ridges behind the clithrum, indicated by the white arrow. They also have a single gill opening, which I mentioned before, directly behind the coronet, instead of the 2 separate gills found in non-pygmy seahorses, as indicated by the red arrows. Here is a CT scan of Hippocampus pontohi, which is housed in the fish collections at the Australian Museum. As you can see, the head, trunk, and tail are heavily ossified, just like the non-pygmy seahorses. However, a closer look at the neurocranium on the right reveals some major differences from the non-pygmy seahorses. First of all, the SOC and the anterior nucleoplate, designated by A and B, are present. However, the anterior nucleoplate does not exhibit a bony structure in the formation of a coronet. There's simply no coronet, it's just a bony plug. As well, the posterior nucleoplate that you see in the non-pygmies is absent here. Lastly, the clithrum is continuous versus discontinuous in the non-pygmies. So it forms a solid collar of bone from side to side, and I think this bony color supports the soft tissue of the single gill opening that you see in the pygmy seahorses. And here's a dorsal view of the same specimen, Hippocampus pontohi, which focuses on the 2 pair of wing-like extensions that you see on the anterior trunk. In this case, the paired spines are similar in appearance and extend laterally from the body. All free-living pygmy seahorses have these wing-like spines, so it's a good character to look at. In similar fashion, here's a CT scan of the paratype Hippocampus culmini from the type locality Lord Howe Island and is housed in the fish collections at the Australian Museum. Hippocampus culmini share similar features with pontohi. As you can see, the head, trunk, and tail are heavily ossified and exhibits a similar arrangement of neurocranial bones. Like pontohi, the anterior nucleoplates does not exhibit a coronet. It just looks like a plug of bone. As well, in the species, the second pair of wing-like spines consist of double spines versus single spines in pontohi. So I have a running bet with a friend of mine who's convinced that Hippocampus culmini and pontohi are the same species. However, the morphology of the paired spines on the trunk distinguish them quite readily. So on to the soft coral pygmy seahorses. This second group are from the Indo-Pacific and are found only in soft coral. Here's Hippocampus satomiae and Hippocampus wellinanus, both from Indonesia and were described in the 2000s. They're habitat specialists found in oligate-associated soft corals. You only find them in soft coral, but different species, around 20, 30 meters. Hippocampus satomiae on the left is the smallest of all seahorses, measuring at just 12 millimeters in length and are nocturnal. In contrast, Hippocampus wellinanus is diurnal. They're very similar in appearance to the free-living pygmy seahorses and having a single gill opening directly behind the coronet, and also appear to have wing-like structures on the anterior trunk ridges behind the clythrum. The coronet, however, appears to be a double coronet. You can see, I mean, it's hard to see, but you can see it's almost like it's a split. So what's going on here? So here's the CT scan of Hippocampus satomiae. Unlike the free-living pygmy seahorses, you can see on the left that there's reduced ossification in the trunk area. And a closer look at the neurocranium reveals some major differences from the free-living pygmy seahorses. A and B, highlighted in green, show that the SOC is disc-like in shape, and the anterior nuclear plate is tooth-like in shape, and they're not connected to each other. So this is quite different. I haven't scanned Hippocampus wallenanus yet, just because it's not available, but I'll get to that. The SOC and the anterior nuclear plate in this species is quite different from any other seahorse I've looked at. And like the free-living pygmy species, there are two pairs of wing-like spines that you see here at E. And the third group, the Gregorian coral pygmy seahorses, Hippocampus bargibandi and Hippocampus denise, they occur in the central Indo-Pacific. They're the first and second pygmy seahorses to be described, respectively, in the literature. Hippocampus bargibandi was described in 1970 from New Caledonia, and is the largest species of pygmy seahorse recorded, attaining a length of up to 2.7 centimeters. Both species are habitat specialists and are found in obligate association with certain species of Gregorian corals at 20 to 100 meters depth, and once settled on a Gregorian coral, they never leave it. So based on their appearance, relative to the other pygmy seahorses I've just described, I consider them to be complete oddballs of the family. They both look very different in that they have bumpy flesh. They look incredibly cryptic since their bumpy flesh resembles the closed polyps of the Gregorian coral in which they are found. It's also assumed that these two species have one gill behind the coronet, but there's no mention of the gills in the original description nor in subsequent descriptions. Phylogenetically, Hippocampus bargibandi and denise are grouped together and are recovered as sister to Pontohi and Colmani. So this is a CT scan of the paratype of Hippocampus bargibandi from New Caledonia and is housed in the fish collections at the Australian Museum. Unlike the heavily ossified free-living pygmy seahorses, the trunk exhibits reduced ossification, just like in Hippocampus satomiae. A closer look at the neurocranium on the right reveals some really interesting differences from all other pygmy seahorses. The SOC is massive. It's just a huge block of bone and is much larger in size compared to the other SOCs and the other pygmies. The clythrum is discontinuous, a trait shared with the non-pygmy seahorses. And then in D, it's hard to see here, but the posterior nuclear plate is present, again, a trait shared by the non-pygmy seahorses. Other differences include the absence of paired wing-like spines and a massive buttress-like pectoral fin base. Here's a dorsal view of Hippocampus bargibandi, and C shows the posterior nuclear plate. I can tell you right now, I never thought I would be so excited about a posterior nuclear plate, but here we are. It just reveals a lot about the species. And similarly, in Hippocampus denise, this specimen is housed in fish collections at the Australian Museum as well. This shows reduced ossification of the trunk, just like Hippocampus bargibandi. A closer look at the neurocranium on the right shows, again, some really interesting features. The SOC is very smooth, with no dry bony formations that you see in the other pygmies. B, the anterior nuclear plate is smooth as well, with no dry bony formations. The SOC and the anterior nuclear plate are aligned evenly with each other. And again, just like Hippocampus bargibandi, the clythrum is discontinuous, another feature that is shared with the non-pygmy seahorses. So these two are very different from the others. Oops. So before I've described all pygmy seahorses having one gill opening behind the coronet. Well, it turns out in both Hippocampus, Bargabani, and Denise, they had two gills, just like the non-pygmy seahorses. In Hippocampus Bargabani on the left, you see them widely spaced apart behind the coronet, which is right here. And then on the right is Hippocampus Denise. Here's the coronet. But here, the two gills are spaced quite close to each other, almost forming one gill. This is because the clithrum in Hippocampus Denise is not quite fully discontinuous. It's almost closed. So this is a huge discovery for me, and this was done at the Australian Museum a month and a half ago. So it's really an exciting discovery. So in summary, for this section of the talk, I've shown the morphological differences in the neurocraniums of specimens that represent the two seahorse lineages, which help to reconcile the phylogenetic data. I also have shown the morphological differences that are present among the pygmy seahorses. I will propose retain all non-pygmy seahorses in the genus Hippocampus. And additionally, I'll propose splitting the pygmy seahorse into 3 genera, using for now the retainer names genus A, B, and C, to accommodate the morphological differences among the species. Genus A will include the free-living pygmies. Genus B will include the soft coral pygmies. And genus C will include the Gregorian coral pygmies. So the revision of Hippocampus is finally happening based on all these morphological characters. All right, so up to now, I've talked about the neurocranial differences between the non-pygmies and pygmy seahorses and the proposed genetic diversity with the pygmy seahorse lineage. On the flip side, I'll now focus on the non-pygmy seahorses and the hidden diversity within that lineage. I'm going to talk about 2 really unusual species that don't quite conform to the typical seahorse. This is unusual seahorse number 1, Hippocampus paradoxus, a seahorse that was described from Esperance, Western Australia. Like all seahorses, it has an angled head, middle brood pouch, and prehensile tail. It can be distinguished, however, from all congeners by the presence of a series of weird, fleshy, fin-like lobes along the dorsal midline of the trunk and tail, as you see there in the female specimen on the left, and the absence of a dorsal fin. The non-type, which you see on the right, is what it would normally look like. You can see the unusual large helmet-shaped head and fin-like lobes on his trunk and tail. This specimen was fortunately found fixed in ethanol after the description of the species, so fortunately genetic data was obtained for this individual. This is unusual seahorse number 2, the very similar Hippocampus minotaur, described from Eden, New South Wales. Again, as with all seahorses, it has an angled head, male brood pouch, and prehensile tail. This species doesn't quite have the helmet-shaped shed nor the fleshy fin-like lobes, but it does have a dorsal fin. A and B on the very left is the pregnant male holotype, freshly collected and preserved respectively. You can see the brood pouch is heavy with eggs and is located on the tail, unlike on the regular typical seahorse, which is on the trunk. So here's CT scan. Oh, sorry, I'm gonna go back here. You can see the brood pouch on the left in the male holotype. The brood pouch is pushed far enough anteriorly to such an extent that the abdominal organs are greatly displaced and the brood pouch appears to be on the trunk. Our morphology of Hippocampus, minotaur, and paradoxus make determining their relationships to other seahorses quite challenging, especially considering the location of the brood pouch. At this point, we need to internally look for clues as to what's going on morphologically with these unusual seahorses. And thanks to CT scans, here are CT scans of Hippocampus minotaur. It's the male on the left and female on the right. Right away, I see morphological characters that are not congruent with those typically seen in either non-pygmy or pygmy seahorses. For starters, I see these really long bony processes protruding from the last trunk rings in both sexes. They just don't appear in any seahorse species I've looked at. In the male on the left, they appear to support the brood pouch eggs below or perhaps the organs above. In the female on the right, the bony processes are present anteriorly and posteriorly on the trunk and appear to support the eggs. So let's look closer at what's going on with the neurocranium. So this is CT scan of Hippocampus minotaur, of its head, where right away I see unusual features. A, the SoC is highly derived and elevated with the tip of it swept back. It's massive. B, I see a remnant or vestigial anterior nuchal plate, which is really weird. And D, I also see a bony ridge on the first trunk ridge, which conforms to pygmy pipehorse morphology. All these features are not congruent with the neurocranial morphology of pygmy, non-pygmy seahorses. I also see a continuous clythrum at C and heavily buttressed pelvic fin base, both of which are indeed shared with the pygmy seahorses. Luckily, we were able to obtain nuclear data from a tissue subsample of Hippocampus paradoxus, which is at the South Australian Museum, from which we can infer the phylogenetic relationships of this species. So you see here in the tree, Hippocampus paradoxus falls in with the other pygmy seahorses or pipehorses. So in the tree, Hippocampus paradoxus is recovered with the Indo-Pacific pygmy pipehorses and a sister to the genera Syngnathidae, Cyclics, and Idiotropicus. So this has clarified some things with this weird seahorse. The Indo-Pacific pygmy pipehorse genera Syngnathidae, Cyclics, and Idiotropicus are all superly similar in appearance. Syngnathidae occurs throughout the Indo-Pacific, including Australia. The closely related Idiotropicus is endemic to Australia, where Cyclics is a new genus from temperate New Zealand, and the description of it is currently in press. So let's look at the CT scans of these specimens to look for shared morphological characters with Hippocampus paradoxus. So here are CT scans of all the specimens that we just saw, and the overall skeletal anatomy of these guys are pretty much similar, except for Hippocampus paradoxus. The male specimens all exhibit these multiple bone extensions that extend ventrally from the anterior tail rings, exactly where the male brood pouch is located. In contrast, the bone extension of the male of Hippocampus paradoxus extends from the last trunk ring, even though the brood pouch is located on the tail. So right away, you see there's some major differences among these pygmy pipehorses, but we need to look at the neurocranium to see the other differences or shared traits. So here's a CT scan of the neurocraniums of the specimens we just looked at, of their heads. I see 4 morphological characters in and near the neurocranium that are shared among these pygmy pipehorses. 1, the bony ridge on the first trunk ring, we see in blue arrow. They all have that in common. 2, the absence or remnant of a posterior nuchal plate. So in the first 3, the posterior nuchal plate bone is missing, but in paradoxus, it's vestigial. 3, a highly derived SoC as indicated by white arrow. So Syngnathidae, Idiotropicus, and Cyclics all have these really interesting bony formations. In Syngnathidae and Idiotropicus, they look like bony ridges. In Cyclics, it's pretty much a cleaved cup-like structure. And in Hippocampus paradoxus, it's a fortified tripartite bony ridge. So in conclusion for this segment, I'm going to propose removing Hippocampus minotaur and paradoxus from the genus Hippocampus and place them into a new genus of pygmy pipehorse that is endemic to southern Australia. So that is a really nice surprise. So in conclusion, I'm going to come full circle. Which genus or species is directly sister to Hippocampus? Well, it turns out, based on unpublished data and on 1,000 genomic UCEs, it's Hippocampus macrorhynchus and Hippocampus punctatus. And that was quite a surprise to me. Hippocampus macrorhynchus is a sand coral dwelling pipefish with a long snout. It's one of the more beautiful ones. And never would I thought that it would be directly sister to Hippocampus. But if you look at CT scans of its neocranium, there's some clues as to why this species shares some morphological characters with the seahorses. On the bottom is Micrognathus crinitis. It's a pipefish from the Western Atlantic, but it represents most pipefish species I've looked at based on CT scans. If you look at the SOC anterior and posterior nuchal plates, they're all fused together. They're all lined evenly with each other. It's classic pipefish neurocranial bone architecture. On top is a CT scan of Hippocampus macrorhynchus. You can see that the SOC is highly derived, and as well as the anterior nuchal plate, but the SOC and anterior nuchal plate are leaning against each other, forming a space between them, just as you see in all seahorses. So perhaps the ancestors of the seahorses and the genus Hippocampus share some traits before they split off. All right, thank you guys. That is it.